Hello, everyone, and thank you for tuning in today. My name is Cooper Mellema, and I'm from the University of Texas Southwestern Medical Center, Department of Bioinformatics. I'm in Dr. Albert Montillo's lab, Deep Learning for Precision Health Lab. Today, I'm going to talk to you about architectural configurations, atlas granularity, and functional connectivity with diagnostic value in autism spectrum disorder. Now, as my work builds off of experiments I presented at ISBI last year, I'll briefly review those experiments for background, go over autism spectrum disorder, the materials and the model evaluation procedure we used. And then I will go much more deeply into the analysis of the models we trained, including their hyperparameter optimization, the learned features, and significance of atlas granularity. So, autism spectrum disorder is a severe psychiatric disorder affecting 1 in 54 children. It's characterized by abnormal neurological development, and currently the diagnosis of autism spectrum disorder is a subjective process that takes a lot of time, skill, and expertise. So a more objective test with MRI biomarkers is of great interest. Furthermore, autism spectrum disorder is largely believed to be a disease of brain connectivity. Therefore, we use multi-contrast MRI, both functional and structural MRI, to quantify the alterations in ASD and then build a predictor. So our goals here are to look at the models we generated previously, look at what type or combination of feature types are optimal in these models, what machine learning model makes optimal predictions under what conditions with these features, what hyperparameters are most suited to the high-performing models, what are the learned features, what are the characteristics of those learned features, and how do those learned features compare across atlases and atlas resolutions. So we do this by building diagnostic models to predict healthy control versus ASD from that MRI recording. So we use 915 subjects from the IMPACT database. This includes derived features per region of interest rather than raw images. This ensures data shareability and greater patient privacy. So going into how we derive these features, given the original fMRI time series, then the brain is parcellated and a connectivity metric is calculated between the regional average time series, and a connectivity matrix is calculated from comparing those time series per region. So we use 7 different atlases to parcellate these regions, and then the metric we used for comparison is something called tangent space embedding. It is a metric that's analogous to something like Pearson's R, but it better captures subject-specific correlations. We also have volumetric information that we integrate as well. So we did a random search over 50 different model configurations per machine learning model tested, evaluated with threefold cross-validation, error into the ROC curve. We looked at multiple shallow or classical machine learning models with nonlinear characteristics, including random forest derivations, boosting derivations, and so forth. And then we additionally used linear classical machine learning methods with different regularization techniques. Finally, we used deep learning models, including a dense feed-forward network, a bidirectional LSTM, and a graph convolutional network. Now, our goal here was to, among these shallow nonlinear machine learning models, shallow linear machine learning models, and deep learning models, to similarly optimize them such that we were able to make an apples-to-apples fair comparison across them all. The random search in particular is a well-validated technique for hyperparameter selection and unbiased selection across different hyperparameter spaces, which might vary greatly in dimensionality. So I'm going to briefly reintroduce you some results I presented last year. In this table, you see model categories shown from left to right, and then the features, functional and structural, listed vertically, and the performance measured by the color bar on the right. Now, we're going to look just at the high-performing models, so we can see that the dense feed-forward network using the BASC atlas with 122 ROIs performed the highest out of all the models tested. So we achieved 80.4 area under the ROC curve, comparing favorably to top impact challenge entrants reported at MICAI. We additionally compare favorably to results on other large data sets such as ABIDE, which achieve up to 73.3 area under the ROC curve when using multiple sites. So now that we have these models, we wanted to extend them. We want to look at the hyperparameter search ranges to see if we had sufficiently broad ranges to isolate performance configuration maxima. We further examined the learned features, looking at which ones are the most important, what is the significance of those, and finally, we did this by looking at hyperparameter analysis through kernel density estimates of the hyperparameter search space and permutation feature importance. So the models we selected to go look at much more depth included the BASC atlas at 3 different resolutions: coarse, medium, and fine, because these had the highest performance across multiple atlases and resolutions. So the BASC atlas is defined by resiliency of clustering with repeated subsampling. We compared the coarse, medium, and fine resolutions again, and you can also see at the bottom that these parcellations are not perfect subsets of each other. There are slight differences among the parcellations. Now I wanted to get a little bit more into how we did our hyperparameter search analysis. We looked at hyperparameter densities for high and low performing models, looking for performance densities in hyperparameter space, comparing the top 20% of the highest performing models as evaluated by validation area under the ROC curve, plotted versus the bottom 20%. Furthermore, we use permutation feature importance, wherein we randomly permute features across all subjects and look at the decrease in prediction accuracy as we destroy the information present in that feature by mixing it among subjects. Then we average this over 16 different repetitions. So here we examine the kernel density estimates for high and low performing models. We can see that the high performing models in blue tend to have fewer hidden layers and less neurons per layer. As we get finer and finer detail, the number of layers in the probability density of where high-performing models were likely to occur, number of hidden layers continued to go up, and the numbers in the first layer went down. So over time, these models became more narrower and deeper. So from this hyperparameter search, we can conclude several things. First, we can conclude that we used adequate search ranges. So the values for the high-performing models tended to lie towards the center of the search ranges and did not abut the edges of the search spaces. Different model configurations were found between high and low-performing models. There was no overlap between those peaks. And the highest performing models used between 2 and 4 hidden layers, 16 and 64 neurons per layer, which is generally very simple for a dense network, but the exact parameters per model are granularity dependent. There is, again, that slight trend of deeper and narrower models as the granularity became finer and finer. So next, we want to look at those features. We're doing this permutation feature importance again. What features did the model learn? And then looking at the importance of the top model across different atlases. So here we have the top identified features for the coarse atlas, the medium atlas, and the fine atlas. We have depicted in the color scale standard deviations from mean feature importance. So the z-score of these different components, and these are putative biomarkers. So we see, pardon me, a preponderance of features that involve cerebellar structures across multiple atlases and particularly several connections such as cerebellar to motor or supplementary motor regions show up multiple times across these atlases. You can see involvement of language areas more laterally in the brain that are common across multiple atlases, these motor areas, cerebellum, and across several of the atlases, frontal cortex as well. I'm going to go into these in quite a bit more detail in the next couple slides as well. So I'm going to orient you before showing you some rather complicated figures. We are going to look at the functions of these identified regions. The top 15 features sorted by z-score importance, standard deviations from mean feature importance, are going to be depicted in a color-coded bar graph where the color code depicts the types of regions connected. So from motor region to motor region, that bar will be colored blue. The length of the bar will be the relative feature importance. Language to language red, non-language non-motor to non-language non-motor yellow, and intermediates on that color wheel as well. So this feature map depicts several patterns. We can see that these features, we can see language cortex featured prominently, cerebellum featured prominently, language cortex again featuring prominently, and then motor cortex also showing up repeatedly. So these features corroborate features previously reported to be altered in autism spectrum disorder. We can see motor and supplementary motor connections in particular, going by that color scale, showing up in this bar graph quite a lot. We know that autism spectrum disorder has sensorimotor processing alterations. And furthermore, we identify these novel cerebellar features that seem to show up again and again. So looking at moving to a finer atlas, we can see that the preponderance of features moves somewhat from motor towards more language. You see these purple bars, language. So we see a slight shift from largely motor-related regions towards language regions. We know additionally there is language processing alterations in the pathophysiology of AST. Again, these cerebellar features show up. And then if we move even finer, we can see that these additional regions that have been known to occur in ASD as well. We see involvement of the frontal cortex more prominently, the prefrontal cortex, and additional regions of association cortex, and non-language, non-motor cortex in general. So looking across these different atlas resolutions, we can say that there are some consistent regions that are important, including those motor, language, and cerebellar features. And then we can also see differences across these resolutions. Again, that shift in preponderance of features, low resolution motor, medium resolution, more language features showing up, higher resolution, we see more and more association cortex. So this study has some limitations, of course. This data set only has binary diagnoses, and autism spectrum disorder is a spectrum disorder. We could additionally have used additional methods beyond permutation feature importance. And then while this data set is intrinsically multi-site, we could additionally validate this on other data sets to further confirm these results. So in this work, we did a systematic search across a wide variety of combinations of feature types and models. We then mined these models for information from their hyperparameters, revealing what kinds of hyperparameter configurations are important for making this diagnostic decision. We confirmed some previously implicated biomarkers. And then we additionally identified some new biomarkers, especially the cerebellar biomarker that occurred across multiple granularities, bolstering confidence that this is indeed a new putative biomarker. Furthermore, we can characterize the granularity at which these performances were achieved. The fact that we found different features and different performance or achieve different granularities suggests multi-scale approaches might be worthwhile in the future. Again, we look forward to extending this work with further development to additional regions, testing, and translation. We have a paper in development, and I wanted to thank you all for coming, or tuning in rather, and thank my PI, Albert, and the rest of the lab, and then my funding sources. Thank you very much, and if you have any questions, please feel free to email me at the address listed there. Thank you again.